The overall goal of this biofilm formation and assessment protocol is to determine the ability of enteric bacterial pathogens to form a biofilm, especially in conditions that mimic gastrointestinal transit and exposure to bile salts.
The methods can help answer key questions in the bacterial pathogenesis field, such as the ability of enteric pathogens to survive the harsh bile salt conditions of the gastrointestinal tract. The main advantage of the techniques is that the combined results from the 3 assays support in-depth characterization of biofilm formation in response to bile salt exposure for each pathogen.
We first had the idea for the combined methods given the need to fully quantify bile salt-induced biofilm formation in Shigella flexneri. Generally, individuals new to this method will struggle because aspects of the assay can limit reproducibility, so please pay attention to the critical steps to ensure reproducibility.
Demonstrating the procedures will be members from my laboratory, Kourtney Nickerson, postdoctoral fellow, and Alejandro Yanez, clinical research fellow.
First, label 2 1.5 milliliter tubes as TSB and TSB plus BS. Then add 1 milliliter of TSB or TSB plus BS to the respective tubes. Next, inoculate the tubes with 20 microliters of overnight culture at a 1:50 dilution. Then, add 130 microliters of uninoculated control media to each of 3 wells in a sterile, clear, flat-bottomed, tissue culture-treated 96-well plate. This will be the blank control. Add 130 microliters of inoculated culture into each of the 3 wells. After